Thank you for this opportunity to present an update on the Horizon clinical trial by Ionis for multiple system atrophy. We're testing a new drug, an investigational medicine called Ion 464. This is my disclaimer slide. A little bit of background about Ionis. Ionis was founded in 1989, and our headquarters are located in Carlsbad, California. And we're a leading innovator in RNA-targeted medicines. We're made up of world-class scientists and leaders, and we really have a passion to deliver medicines to people who need them the most with severe conditions. And our medicines are antisense oligonucleotides, and I'll explain what that means. And they're designed to disrupt the disease process. And this has led to the development of 5 approved medicines to date, and we have many more investigational medicines in development. I wanted to walk through how antisense oligonucleotides work. So the first place to start with that is understanding how proteins are made, because our medicines work directly on protein levels primarily. So this starts with DNA, which you can see on the left-hand guide. DNA is the genetic, on the left-hand side rather, DNA is the genetic material in your cells. And it's kind of like a master cookbook. So there are a lot of recipes that make all the proteins in your body, and each protein has its own recipe, which is called a gene. And the DNA is kind of like the cookbook that contains all those recipes. And proteins are tiny molecules that are required for everything in your cells, for your cells to live and to work. And when your body needs a protein, the DNA partially unwinds, and then that exposes the recipe for the protein. And this causes a temporary code of the recipe to be made, this DNA unwinding. And this copy is called mRNA, which stands for messenger RNA, and that's pictured here in the middle. And there are specific parts of the cell that read the mRNA and then use this information to produce the protein. So the technology that we produce at Ionis, antisense oligonucleotides, or ASOs for short, is a type of drug that acts on this RNA temporary copy to affect the amount of proteins that are made. And ASO technology has the potential to target diseases with genetic components and causes. So as the ASOs act on the mRNA, they can either increase or decrease protein production, and they can also change a protein that's produced. So here in this picture, you see what happens when an ASO interacts with the cell. It enters the cell, it's the pink strand shown, and it enters the cell, and then it binds to the mRNA. And this triggers an enzyme to work, RNase H1. And what RNase H1 does is it cuts this new duplex that's formed between the ASO and the mRNA. And when that happens, the mRNA is degraded, the mRNA that was supposed to make the protein in question. And so the protein production, in this case, in this image, is decreased. And MSA is a disorder resulting from too much protein or protein aggregation in the brain. And the name of the protein, as you may know, is alpha-synuclein. So Ion 464 is our investigational ASO designed for MSA, and it targets mRNA expression of SNCA, which is the mRNA that makes the protein alpha-synuclein. And so when, as you can see in this image, the SNCA gene is on the left, the mRNA for the SNCA gene is in the middle, and then Ion 464 binds to that mRNA, forms that duplex. The RNase H1 does its job to cut the mRNA, and then the alpha-synuclein protein cannot be made. So our hypothesis is that this will decrease alpha-synuclein aggregation in the brain and also slow MSA disease progression. So let's look at how ASOs are administered to people. So there are different ways ASOs can be administered to people, but when we think about neurological diseases, we need to get the drug into the brain and past a barrier that's called the blood-brain barrier. So this can be tricky, and in order to do this, a lumbar puncture right now is the best way to get the drug past the blood-brain barrier into the cerebrospinal fluid that surrounds the spinal cord and the brain. And so this lumbar puncture involves a fine needle that's inserted into the lower back, as you can see in the picture on the bottom left. And the needle goes into the space between two vertebrae. We collect cerebrospinal fluid, or CSF, in order to look at different proteins like alpha-synuclein in the patient, but we also deliver the investigational medicine through the lumbar puncture as well. So the research and development process takes a long time, and it's outlined in the arrow at the top of this figure in pink, dark pink, orange, and green that you can see here. There's an incredible amount of research that's done before the investigational medicine is ever tested in humans. As you can imagine, first we test multiple ASOs for MSA, and then we design an ASO that's specific to human mRNA for the SNCA RNA. And then we select the one that has the most promising results. Once we do that, that's called the identity lead candidate selection. You can see that in green in the top arrow of the slide. This happened in our research program for MSA in 2017. And then we embarked on a lot more testing in animals, looking at safety primarily and also potential efficacy. And that's standard for all clinical trials. And we also looked at this in models of the disease, such as animal models. And then in this process, we're leveraging Ionis' 30 years of experience in designing and testing ASOs to create Ion 464 specifically. So now in 2024, we're collaborating with expert physicians around the world to carry out the phase 1 Horizon clinical trial. So for the phase 1 is the first step in many stages of the clinical studies. So you can see in the phase 1, the goals as shown here on this slide are to assess safety and tolerability, measure drug exposure in the CSF and in blood, and we also evaluate that for multiple doses, lower doses and higher doses of the investigational medicine. And we do this in multiple countries. Our inclusion and exclusion criteria are shown here. We are selecting patients who are earlier in the disease process. And that's intentional because our experience with other ASOs shows that the earlier we treat, the more likely we are to prevent progression of disease. So we want our clinical trial to have successful results ideally. So we are looking at patients who are a little earlier in the disease process. So they need to be 40 to 70 years old, diagnosed with probable or possible MSA, and that can be cerebellar or Parkinsonian type MSA. They do need to have brain imaging consistent with neurodegenerative Parkinsonism, and they do need to be able to walk at least 10 meters. We are excluding patients who have cognitive dysfunction and a known family history of ataxia or Parkinsonism. We're enrolling currently for our phase 1 in these 5 countries. In the UK, we have site in London and in Oxford. In France, our sites are in Paris, Lyon, and Toulouse. In Austria, in Innsbruck. In Germany, we have 4 sites: Dusseldorf, Hanover, Marsburg, and Ulm. And we have 1 site in Portugal near Lisbon called Lourdes. So that's the update on the MSA clinical trial Horizon by Ionis of Ion 464, an antisense oligonucleotide. Um, Thank you to all the patients and families who are participating in this trial and to all the expert sites as well.